Hello everyone, my name's Luke Odell and I work at the Institute for Frontier Materials at Deakin University, Australia. And in this talk I'm going to tell you about some of the work we've been doing setting up magnetic resonance imaging experiments to study thermo-electrochemical cells functioning in situ. So what are thermo-electrochemical cells or thermocells? Well, they consist of two parallel metal electrodes and an electrolyte that contains a redox active species. And what happens is we get redox reactions occurring within the electrolyte, and the equilibrium of this redox reaction will depend on temperature. So the balance at each electrode is different, and this leads to transfer of electrons through the external load. So what we get typically is reduction occurring at the hot electrode, oxidation occurring at the cold electrode, and a continuous circulation of the different redox species. So this basically allows the devices to generate continuous electricity under the application of a thermal gradient. So you could, for example, attach these devices to a hot pipe and generate electricity from the waste heat from that pipe, or equally from a human body. Now these devices don't generate a lot of electricity, but there's no consumption of any species and no plating or intercalation or formation of any interfacial products that can cause problems in devices such as batteries. So these devices essentially give you free energy. They should have an extremely long lifetime, and so they're worthwhile investigating. Now, despite the simplicity of the concept, the processes that are occurring inside these devices when they're working can be extremely complex. So obviously you have the applied temperature gradient, you have redox reactions occurring at both electrodes, you have exchange of the counter anions between the two different types of cations. You're going to end up with spatially dependent concentrations of the two different redox species. And you may or may not have thermal convection occurring depending on the relative orientation of the thermal gradient with respect to gravity. So for example, if you have horizontal electrodes with a hot electrode at the top, you wouldn't expect any convection. The species will also undergo diffusion, and thermophoresis can also occur, which is where the thermal gradient itself can lead to concentration gradients, with the larger species clustering on the cold side of the device. And it's important to know that all of these factors will be interdependent in complex ways. And they'll also be sensitive to the cell size, the geometry, and the orientation. And if the temperature gradient is changing as a function of time, or the load resistance is, then these factors will also be time dependent as well. So although the design and the way the devices work is relatively simple, there are potentially really complex things that can occur inside them, and this is something that we could look at potentially using magnetic resonance imaging. So there have been a number of papers that have used numerical modeling to visualize some of the processes that occur inside these devices, such as concentration profiles for the equilibrium device between one electrode and the other, things such as convection, temperature maps, that kind of thing. Now, MRI is a prime candidate for obtaining experimental verification of these types of models. For example, MRI can measure transport of individual species via diffusion tensor imaging. We can observe mass transport via velocity mapping. And we can try to look at concentrations of the paramagnetic species via relaxation contrast. And obviously, since we have paramagnetic species in here, that's going to give us significant relaxation enhancement and is going to be a big potential advantage for the MRI contrast. The T1 relaxation times are also obviously going to be sensitive to temperature. And the dependence of these T1 relaxation times on both the sample temperature and the concentration of the paramagnetic species is an important point that I'll come back to later in the talk. So this is the specific electrolyte system that we're studying in this project. The main component is the cobalt tris bipyridine species, where the cobalt can undergo redox reactions between the +2 and the +3 oxidation states, and it's the +2 state that is paramagnetic. The counterion is this NTF2 or TFSI anion that you might recognize from the world of ionic liquids, very popular anion, and the solvent we're using is MPN. And we're looking at both the ordinary liquid form of this electrolyte. We're also looking at a gelled version, which we make by adding 5 weight% PVDF. Now, this system was chosen based on its Seebeck coefficient, which is a measure of the amount of potential generated per unit of temperature differential. And this particular value has the highest Seebeck coefficient yet reported. And this was reported by our group, and our group is also currently working on tailoring these ligands around the cobalt to push this value even higher. So protons are the obvious choice for MRI, although I will note here that we're also using fluorine-19 imaging, but I'm not going to show any of those results today. So this is what the proton NMR spectrum looks like from this electrolyte. You can see that the spectrum is dominated by the signal from the MPN solvent, and we can also see smaller peaks here from the cobalt ligands, clustered around 10 ppm for the non-paramagnetic form, and then spread out over much larger shift ranges for the paramagnetic cobalt 2+. Now, while these paramagnetic shifts for these protons opens the possibility of shift-selective imaging to observe the paramagnetic species, they do, as you can see, have very small intensities, and obviously they also have very short relaxation times, so they're not really amenable to MRI. So what we're doing to record our images is actually focusing on this MPN signal. And this allows us to probe the paramagnetic species indirectly. So this image shows a T1 map of a phantom that we made up using several different samples in 5mm NMR tubes. So this is just an axial slice, and it was obtained using the rare imaging pulse sequence, with images obtained at different recycle delays and then converted into a T1 map using the Prosper software. So as I've said, the signal that we're observing arises from the MPN, and clearly you can see here the MPN proton T1 is very sensitive to the different concentrations of both the cobalt 2+ and cobalt 3+ species. So this is essentially a proof of concept that we can use T1 contrast MRI to obtain T1 maps from a thermocell. So the next thing we needed to do was design and build the cell itself so that we could actually set up the device inside the magnet. Now, those of you who know anything about NMR or MRI will know that it's generally a very bad idea to bring metal objects close to a superconducting magnet. And this is particularly bad for imaging due to the significant distortions that can result. So this image, for example, was obtained by somebody with metal braces in their teeth. And since we want to image processes inside our thermocells that are happening very close to the metal electrode surface, we need to find a solution to this problem. Fortunately, it's quite simple for a cell design such as this, because if the metal electrodes are parallel and flat, it's possible to largely eliminate these distortions by aligning the surface of the electrodes with the RF field. And this can allow you to obtain images from the electrolyte right up close to the electrode surface. And that's where the reactions are happening inside our thermocells, so that's really important. So after a few iterations, these are the cell designs that my PhD student Isuru Gunathilaka came up with. So they involve the use of two gas lines to control the temperatures on each side of the cell independently. So the hot and cold gases flow in and out through the exterior of the cell, and they flow past the exterior sides of the electrodes, which are just platinum strips, and they're held in place by rubber gaskets. The cells themselves are made from the PEEK polymer, which is unreactive and fairly easy to machine, and it's commonly used for cells for in situ NMR and MRI experiments. Then we have obviously wire connections to the platinum electrodes that allows us to connect these to an external potentiostat. And the hot and cold gas supplies are coming from two BCU chiller units. So this design allows the temperatures of each electrode to be controlled independently, and it allows us to establish a stable and controllable temperature gradient across the electrolyte. The design for the horizontal electrode version is modular and can be set up with a temperature gradient in either direction, so the hot electrode at the top and the cold electrode at the bottom, or vice versa. Though I should note that the hot gas always needs to come from below, and that's because we're using the heater inside our microimaging probe, which is located below the cell, to heat the hot gas. So this modular design allows us to apply that temperature gradient in either direction despite that limitation. The trickiest part of this particular design is getting the electrolyte into that central chamber so that it makes good contact with the top electrode without any bubbles present, as bubbles can really wreck the images. So this is the overall setup that we have to run the MRI experiment on a working thermocell. It's quite simple, but it does take some skill and care to get the cell into the magnet with all of the connections in place. I offered to help the student Isuru do this, but he's very independent and he rigged up a system of pulleys and string to be able to do this on his own. So you can see we have the two separate gas supplies for the hot and cold gas, and we have the connection to the potentiostat coming through the top with an RF filter in place and RF shielded wires. So these images show temperature maps that we obtained from the vertical electrode cells with a liquid electrolyte on the left and a gel electrolyte on the right. So these were generated just from the T1 maps, again obtained with the basic rare imaging pulse sequence. And in this case, the electrodes were unconnected to an external load, so there were no redox reactions occurring. And this allows us to assume that the concentrations of the two different cobalt oxidation states are uniform. And this means that the T1 value for each pixel in the image can be easily converted to a temperature via calibration measurements that we'd already made. So then knowing the temperature of the electrolyte as a function of space is essential to later on generate the concentration maps of the redox species in the working connected cell. The temperature difference between the hot and cold electrolytes in these images is 90 degrees in both cases. On the left we can see a more or less uniform temperature across most of the electrolyte, with colder and hotter regions visible at the electrode surfaces. This is obviously due to thermal convection that rapidly circulates the bulk of the electrolyte, and you can actually see a cooler kind of stagnant area in the center. For the gel electrolyte, the convection is prevented by the polymer matrix, so what we end up with is a linear temperature gradient between the two electrodes. So the next thing we want to do is connect the electrodes to the external load and try to observe the resulting redox reactions in the form of concentration maps. The problem here is that the measured T1 value for each pixel will depend on both the temperature at that point and the relative concentration of the redox species. Fortunately, we already have the temperature maps from the unconnected cells, and we assume that the spatial variation in temperature is unaffected by the redox reactions once we connect the electrodes. We can then use calibration measurements to determine an empirical formula for T1 as a function of both the sample temperature and the concentration of the paramagnetic cobalt species. And this surface then allows us to convert the measured T1 for each pixel into a cobalt 2+ concentration using the known temperature for that pixel. So this is a cobalt 2+ concentration map obtained from a liquid electrolyte in the vertical electrode cell. The electrolyte contains 0.05 molar concentrations of both redox species. And the temperature difference between the two electrodes in this case is only 20 degrees C. The reason for this was to not have too much convection occurring inside the cell, which can potentially lead to artifacts and image distortions. Nonetheless, there will be some convection occurring in this cell. And one avenue we're pursuing is to use velocity mapping experiments to measure the convection, but I won't show any data on that today. So this particular image was obtained from a cell operating under a 30 kiloohm load, and you can see a clear concentration gradient in the cobalt 2+, with higher concentrations at the hot electrode as we'd expect, while in the center the concentration is close to 0.05 molar. Strangely, in the gel sample, we actually saw the reverse of what we expected, with cobalt 2+ concentration very high on the cold electrode side. And this may be explained by an initial concentration gradient caused by thermophoresis. Thermophoresis results in heavier molecular species moving along a temperature gradient towards the cold direction. So under this effect, we'd expect to see both types of cobalt species clustering on the cold side of the cell. And obviously the relaxation is dominated by the cobalt 2+, the paramagnetic species. We wouldn't expect to see this effect for the liquid sample because we have convection continuously mixing the species. But in the gel sample, we believe over time this can take place, and this is what we're seeing here. After 2 more hours, we do start to see a layer of cobalt 3+ being formed on the cold electrode, as expected, and what looks like some increase in 2+ concentration on the opposite side. So clearly we need to run some time resolve tests on the unconnected and connected cells to differentiate these two processes, but we haven't done this yet on the vertical electrode setup. We do, however, have some time resolved images obtained from the horizontal cell setup. So in this case we're studying the liquid electrolyte, but because we have the hot electrode at the top and the cold at the bottom, there is no convection. The temperature map shows a fairly linear temperature gradient between the two electrodes. And note that the top surface in these images looks a little bit distorted. And this is likely due to the presence of bubbles, which are unfortunately very hard to avoid in this cell. So after connecting the cell to a 5 kiloohm load, we can see the formation of cobalt 2+ concentration gradients that grow over time. You can see that it's quite a slow process and is still changing after 10 hours. And this is due to the lack of convection, meaning that the cell is relying on the far slower diffusion process to ferry those redox species between the electrodes. And this process does correlate also with a continuous slow reduction in the power being drawn from the cell. So just to summarize, I've shown that we can use in situ MRI experiments to obtain quantitative temperature maps and concentration maps from operating thermocells. The paramagnetic state of the redox species leads to significant T1 relaxation contrast, and this is a big advantage for using MRI to study these particular types of devices. And we've seen in these images the build-up of concentration gradients within the electrolyte, and we can map that as a function of time and correlate it with the device performance and other factors such as changes in the external load or changes in the temperature gradient. And it's important to also take into account effects that can occur in these systems such as convection and thermophoresis, which we can also visualize. So finally, I'd like to thank in particular my PhD student Asuru Gunathilaka, who has worked extremely hard on this project. He's shown amazing perseverance to be able to get this working, and he should be finishing up his PhD within the next few months. And also other members of our group, Professor Maria Forsyth, Professor Jenny Pringle, and Dr. Abuzar Tahiri, who have each contributed to the work. And I'd also like to thank the ARC Centre of Excellence for Electromaterials Science, which is funded by the Australian government, for supporting this work. And thank you all for your attention.